He's going to talk about resilience of grasslands under extreme drought and flood. So I think a very interesting topic, and obviously you're brought a big crowd, so
okay,
hand over to you.
Thank you. I hope this is working with the. Okay, so thank you, and I'm very pleased to be here this morning. If anything, because I really want to tell about the incredible amount of works that people working in my team have gone through during the summer. It's been epic. And I hope the pictures I'm going to show will make justice of this incredible amount of work. This is a work based on an SFI grant that I got basically when I get into UCD. It's funded by SFI under the Frontier for the Future program. And it revolves around how grasslands respond to extreme perturbations, especially drought and flooding, especially the combination of the two. It's just no longer about drought per se or flooding. It's the combination these days that is really important, which you can see very well from what has happened during the summer. We had a very dry end of May and June. And the farmers were excited. Wow, we're going to have this incredible hay. And they were actually writing to us, you can't do your experiment because in July we're harvesting. And then all the rain came down in July. And we're very happy about that because the experiments then worked. But it's been a disaster. And then in August, the farmers were struggling, trying to find a couple of days with a bit of sun to cut the hay. And the idea is that now there have been a number of projects in recent years around grasslands, drought, flooding. But they have mostly focused on soil and maybe the soil food webs and general properties of soil. The specific point we are addressing in this project is actually about the microbes living in the roots of plants. And the reason is that they form these very complex networks with plants. And we believe, and that's our hypothesis, that those complex interactions taking place between different plant species and populations of plant species on the one hand, and the microbes on the other hand, this is what mostly regulate the response of grasslands to combined drought and flood. It's a very, very ambitious hypothesis to test. And so we're investigating these in many different ways, mostly in the field, but also with glasshouse experiments. And the idea is that we investigate those networks under a broad range of conditions in terms of floods, drought, and as variable as possible environmental gradients. And so we have a number of questions there. And then we will link those responses of the plant microbial network to things like productivity, for example, of the grasslands or ecosystem respiration, so that we can connect the structure of the communities to ecosystem level functions and properties. And in the summer, we have done this experiment, which took 2 years to set up. In year one, last year, we actually first found the appropriate sites and liaise with the farmers to make sure we would have access to the land. And we did the plant survey to know which plants are dominating the community. And these are the range of sites we managed to secure. So the implication is we had to go to all those places there. We had to set up all those experimental sites that you will see. But the key idea was that in every site, well, actually in every location, we have dry and wet sites. So many of the locations actually are along flood plains, which could cause some problems sometimes because I don't know if you can see that here, but that is one of our sites. It got flooded just when we were about to collect, the experiment was done at that point, but we had to collect all the stuff there. And in fact, 2 of people in my team managed to do that, and I can assure you they are alive and in fact in the audience. So it was very, very entertaining. And that's a very dry place, okay? And then on top of that, we had the rain out shelters. The rain out shelters are supposed, those structures there that you will see a bit better in the next slides, they rain out, they shelter out the rain. So if it's raining, then the rain doesn't go into the soil, and then that can create an extreme bout of drought, especially the season we had. Because you need a very nice initial dry setup, which we had in June. That's just pure luck, right? You just hope this is going to happen, and it happened. And then you need the rain so they can shelter out the rain, which happened as well. Okay, so we are focusing, there are about 80 species of different plants, herbaceous plants across all the sites we look at. But the communities, just because of the type of sites we selected, they are vastly dominated by the by the plant species you can see there. And those are the plant species we specifically sampled at the end of the experiment. And you can see that, already without doing the experiment, places that tend to be flooded have communities that are different from places that are drier. And so we expect that the response to drought would then be very different. That's a picture of, this is me after a good haircut in one of my very rare appearances in the field, I have to say, this year. And this is some of the people involved there. You can see how we are setting up one of the sites. The electric fencing you see there is to keep cattle or sheep out. It's a bit very nosy, and there would be grazing. We don't want grazing happening there. Cows tend to destroy the shelters as well. So with this electric fencing, it worked okay. The only couple of problems we had was that some people at some point stole the solar panel we were using to energize the the fence, but luckily it happened late in the season. So yeah, 120 quid maybe valuable. So you can see more pictures of that, setting up the sites, and then surveying, and taking all the information we need to get out of the soil. And Andrew is a very tall man, and you can see the shelters can actually stand them. They need to be very sturdy and robust. They stay out for 3 months, any type of weather. Should some animals go, it's not easy to put these things up. Took a bit of work. So they stay there for a long time. We have a number of them, and then after 2 months, basically, we went back and sampled. And the first thing you need to know is, did the shelter work? And it did, in some cases. So, in dry places especially, this is very raw data we just got a couple of weeks ago, because we just finished the season, really. We have these probes in the soil that basically monitor soil moisture and temperature every 15 minutes. So they were under the shelter. So you can see in red, you have soil moisture profile. And on the x-axis, that's on the y-axis. And then on the x-axis, you have time, from the time we set up the shelters all the way down until harvest. And you can see how soil moisture drops exponentially, very rapidly under the shelter. So this is when it works. Sometimes it doesn't work that well in flooding areas where the soil was very wet from the very beginning. It's much slower, a bit less clear. It doesn't matter, because in the end, what we want is an extreme gradient with very, very dry and very, very wet places. And the shelters allow us to get that gradient. And the control, it's very interesting. Initially, also the control goes down. And why is that? Because we set up the sites in May, and May and June was very dry. So it actually didn't rain at all, almost. So you see the water in the soil going down, and then in July we have all the rains. So you see the red curve going up. Those are the pulses of rain you get in July. So where the shelter wasn't. So it worked beautifully in some sites. And then we went to harvest and collect the samples. You can see control and drought, where the drought worked, the biomass in a given area of our experimental sites. And we did the dry weight of that. In some cases, the effects are very, very obvious. But what we're really doing, and that was an incredible amount of work, was collecting plants and roots, because we are looking into the microbes in the roots, inside the roots. It's not rhizosphere, it's really the endophytes. So we are collecting all these plant individuals belonging to the A species. And now what we're going to do is we need to sequence those with markers specific for fungi and bacteria, so we can reconstruct the microbiomes inside the roots of the different plant species under different conditions, especially drought and flood, or more or less wet soil, and all the other parameters we are measuring in the soil, which I have no time to go into. And eventually we can reconstruct those networks in which we associate a plant species or a population under certain conditions to their microbes. We have other studies that have already shown the impact of, with the same methodology, the impact of drought on soil biota, but there is very little about what's happening in the microbes in the roots of different species, the species that dominate the plant community. So that's the novelty. And we know from our pilot studies that our molecular techniques are working. For example, here we have this is just a pilot, but you can see that if you categorize plants by different functional categories, for example, grass and forb, which we had in the pilot, the separations in terms of the microbiota, both for 16S and ITS, is very, very good. So just want to drop that there to show that we're actually already collecting data, but it's really the very first stages. This work would not be possible if you don't have a big team with people who really, really willing and really love working in the field and at the same time understand the science. So I just want to say thank you to all the people in the lab. You have done an amazing job, and it does take lots of work and especially good teamwork. So thank you. And this is all of them in the slide. In particular, I want to thank Manon Longepierre and Patrick Doran, because their background work in year one was fundamental to establish the sites, liaise with the farmers, and secure them. It's not easy to do that. So thank you to them. And then finally, also thank you to all the other PIs that are in the projects that I haven't put here, because here I wanted to give space to the early career ones, well, especially John Earsley and Elke Eichelmann, who have been very important in the project as well. And finally, thanks to the UCD Earth Institute, because this project, the idea of this project actually was born thanks to RE-EcoNet, which was a strategic priority project that the Earth Institute has supported. So thank you. And any questions, very welcome.